Great. Well, I already introduced myself, but just to say again, my name's Dan Macqueen. I'm the Deputy Coordinator of AQUA-FAANG based at the Roslin Institute at the University of Edinburgh. I'd like to just, start by welcoming everyone to the training. I hope you get a lot out of it and learn a lot about the bioinformatic analysis of regulatory elements. A special welcome to those from outside the AQUA-FAANG consortium, including Bob Reg and GeneSwitch. I'd also actually like to thank up front everyone who's been involved in building the training and who's going to deliver it. So many individuals from EMBL EBI, NMBU as well, and also Paran from Edinburgh. So I guess my job is probably the easiest one compared to the other trainers. I'm just going to say a few words about AQUA-FAANG, which is, co-delivering and partly funding this training, just to frame the course. And I realize many of you on the training are already very familiar with the project and the broader goal of like FAANG and those sorts of things, but perhaps not everyone. So I think it's an important thing to do. If nothing else, it's a gentle intro to warm you up before Thomas's more hardcore session all about containers today. So in AQUA-FAANG, then, we're working with the 6 most commercially important farmed fin fish in European aquaculture that make up about 90% of the total value of production. We're generating functional annotation data for these species to improve our fundamental understanding of farmed fish genomes, but also to help us be able to predict, using functional genomic information, commercial traits using genetic information, particularly with a main focus on disease resistance, which is a big problem in aquaculture. There's lots of problematic diseases. So we're about 2 years into the project now. This was a 48-month project originally. We're likely, due to the COVID pandemic impacts, to have a 6-month extension to the project. And Sigbjorn Lean, based at NMBU, is the lead of this project and consortium. So AQUA-FAANG in a nutshell. So the current situation is, of course, that we have high quality genome sequences for all the major farmed animal species these days, including most of the aquaculture species, and that's the case for AQUA-FAANG. And we have these 6 species as our focus in the project, which you can read there at the bottom. Of course, generating a high quality chromosome anchored assembly is not as much of a challenge these days as it once was, and getting a high quality annotation of coding genes too, using RNA sequencing. And the whole modern synthesis of animal genetics research is built on using these sorts of resources to interpret GWAS, comparative genomics, that sort of thing. There's much we don't know about how fish genomes are being regulated to influence phenotypes, and it's a big knowledge gap, and it requires new types of functional genomic data, and we're trying to take that step forward in AQUA-FAANG for these major farmed fish species. So I'm not going to go into these objectives of the project in any detail, but just to say we're not only trying to make fundamental knowledge advancement in this project, we're also trying to have commercial impact. And the objectives, generally speaking, are to generate standardized data and also protocols, to share that openly, in addition to spreading expertise in functional genomics in the fish community. Due to the emphasis that we're placing on understanding disease resistance, we're annotating the genome not just in sort of healthy or control states, also immune activated states, and several people here are working on this, of course. And we want to use the functional genomics data to actually help improve the prediction of disease resistance or susceptibility, as well as other traits. And in AQUA-FAANG, we're comparing 6 species that last shared a common ancestor around 300 million years ago. And we're using a sort of comparative framework to help define conserved elements in the genome, as well as lineage-specific elements, which adds a lot to the annotation. And we're also trying to work with end users to help define pathways for them to actually use our data generated in the project for actual positive applications in commercial aquaculture to help its sustainability. So genome functional annotation, well, it's obviously moving past simple predictions of protein coding genes, and that includes a better understanding of the sort of vast universe of non-coding RNA, which has roles in regulating gene expression, microRNAs and other small RNAs and long non-coding RNAs. But that's not really what the training is about this week. This week, we're more interested in gene and genome activity, things like enhancer or promoter elements that are bound to by DNA binding proteins to regulate gene expression under different conditions. And these can be identified using bioinformatic analyses of particular sequencing technologies that have been defined by the Global FAANG initiative and are being used across the three AQUA-FAANG Horizon 2020 projects that are represented here today. So apologies if you've seen this slide many times. So the FAANG initiative is using sequencing assays that I guess were first defined by the ENCODE model organism initiative that can be used to sort of probe the regulation of the genome and gene expression at different physical scales. So on the left, methods like Hi-C and related methods can be used to look at long-range DNA interactions and help define the organization of the chromatin, look at things like promoter-enhancer interactions. So we're generating none of this sort of long-range epigenetic information in AQUA-FAANG, but I believe the other two projects are. But in AQUA-FAANG, we're generating lots of ATAC-seq and ChIP-seq data, as well as RNA-seq data. So you can see they're highlighted in blue there. And you're going to hear a lot more about these methods, of course, but ATAC-seq briefly then, when done well, is looking to identify regions of open chromatin DNA, which isn't bound or wrapped around histones and is accessible to transcription factors and DNA binding proteins, and obviously often represents regulatory elements. And ChIP-seq, which is aiming to capture the interaction of DNA binding proteins with their target DNA, which can be transcription factors, but also histones and their modifications, which Piran will explain later, and that can help us identify and distinguish different classes of regulatory element in genomes. So the project is in a very intensive phase of generating data now, and many participants on this course have been centrally involved in this ongoing enormous effort. This table's a bit dated. Matthew could probably give an update that was more accurate. But we're well underway now in this project. Around 2,000 libraries have been sequenced, which is mostly RNA, and ATAC-seq work has been done, and the ChIP-seq is in progress in the different species. And we've been a bit delayed in the project in generating data, which of course has been due to the pandemic and it keeping us away from fish or from the lab or from reagents needed to complete the work, but we're going well still. And the main functional annotation maps, for those who don't know, that were generated in AQUA-FAANG are really scientifically interesting. So across these 6 different species, we have a panel of the same tissues for both immature and mature fish of both sexes. We have different stages of embryogenesis, from when the genome is activated through to when you actually have a patterned larvae with all the main organs, etc., a little fish that can swim. And also different immune activation states to probe the response of the genome to bacterial and viral challenge. And all the data in AQUA-FAANG is being analyzed through a work package that EMBL EBI are leading, using the same standardized pipelines across the different species, probably the same pipelines you're being trained on this week for ATAC and ChIP. And then that will deliver standardized data back to the project users through an FTP site where different stages of the data analysis can be accessed by the users. So things like the alignments right through to the peak files or the expression counts for RNA data, etc. So then the question is, why is this source of data important for aquaculture? I guess this is to farmed animal biology and production generally. We're well aware that genomes are highly enabling resources for genetics in aquaculture, for genetic management and stock improvement. That's highlighted in this recent review by my colleague led by my colleague Ross Houston at the Roslin Institute, who's a work package leader in AQUA-FAANG. An important question that I think is often and increasingly leveraged at FAANG is, how does genome functional annotation help enhance, farmed animal production? And there's a recent perspective that I guess gives an overview of this sort of thing, that was led by Emily Clark, another colleague based at the Roslin Institute, and this had authors from the 3 different AQUA-FAANG projects on their sort of vision of what called From Fang to Fork, looking to the future of how this sort of data can improve farmed animal production. I think that the sort of classic argument in terms of genetic studies is that functional annotation can enhance our ability to predict variation in traits, perhaps by helping us prioritize, or at least rank causative variants in regions of interest, such as those defined by GWAS, where you have a huge number of markers that might be in linkage disequilibrium with the actual causative variants. Functional annotation data like TAC and ChIP-seq helps us identify particularly variants that are not in obvious locations, like in cis-acting enhancers, which might be like a needle in the haystack. And while it's not proven, I think the idea in AQUA-FAANG, and this is certainly being explored in GeneSwitch and BobReg, is to incorporate this sort of prioritization of functional genetic variants into genomic selection approaches and models, and to see if that enhances the prediction of breeding values, particularly relevant across distantly related populations. And also this sort of prioritization of causative variants or potential causative variants can deliver targets for more precise genome editing, which is increasingly important. But I also don't think that we should forget that I think that the primary reason for generating this sort of data, and it's this sort of stuff that gets me really excited about the data we're generating in AQUA-FAANG. Functional annotation data, especially uncovering regulatory elements, allows us to address really interesting fundamental questions that are also underpinning to many other questions that are applied in farmed animal biology. So how does an animal go from being a single cell to a complex organism with different differentiated tissues and sex differences, effectively using the same genome or set of genes? How do different species generate conserved cellular phenotypes? How do these sorts of features change during evolution? And regulation of gene expression and gene activity is really central to these fundamental problems. And I find that really engaging about these projects. So just important to note too that FAANG projects, including AQUA-FAANG, all sort of conform to fair data principles, that the data should be findable, accessible, interoperable, and reusable. So this in practice means that all the data that runs through these projects has to come with high quality information about the sample origin, as well as the protocols that we use to generate the sequencing data. That's obviously not the standard approach in functional genomics for those used to reading methods in papers, and it's really quite a special thing about the FAANG initiative, in my view. AQUA-FAANG has its own data portal, as do the other 2 FAANG projects. And many involved in this training are currently learning about the fun of getting these metadata submissions through. the last thing to say is that the AQUA-FAANG data will be freely available through Ensembl in its final form. The final genome versions of the 6 species should be available in Ensembl release 105 in the summer. That gives us a suite of tools that we can use, both comparative tools, but also tools for looking at genetic variation. And just to quickly say, there will be a training course later on in AQUA-FAANG on exploiting Ensembl tools for farmed animal research. And I hope lots of people here will be able to come to that course too. So that leaves me really just to say, enjoy the course to everyone, and I think I'll stop speaking there.